Good afternoon, everyone. Thanks for the opportunity to present my work, BVSegNet, the analysis workflow for the blood vessel segmentation. So we know that cancer hallmarks, one of those is inducing angiogenesis. So angiogenesis meaning the formation of new blood vessels so that the tumor can progress effectively. So when you look at the tumor vasculature in terms of normal and the normal vasculature is much more uniform and the morphology is much more uniform. But when it comes to the tumor, it is more chaotic and it's very disruptive. So it is important to study this disruption because we need to treat these patients with radiotherapy and other vascular targeting agents. So that you need the measurement of these blood vessels so that you can treat these patients. So given the radiotherapy, the blood vessels again disrupts in its morphology as well. So it is important to measure them, and then whenever the blood vessels tend to grow more or create more of the blood vessels, what happens is more number of branches are getting created. But when the patient is kind of responding, the number of branches it forms is going to be less. So that is why it is important to measure them. And the traditional imaging modality or the acquisition techniques are restricting towards the ultrasound to see the localized blood vessels. So we wanted a high dimensional and high throughput methodology to counter this effective measurements of the blood vessels, so that we are using digital pathology, which is a high throughput and quite reliable method, using the CD31, which is an endothelial cell based marker, from where you can obtain these blood vessel morphology. So the neovascular density is associated with progression, not only in breast cancer, but also in multiple other cancer types according to the literature. So here we have the breast tumor biopsy that is taken at two different time points. One is before radiotherapy is given to the patient, and 2 weeks after the radiotherapy is given to the patient. So as you see here, the CD31 is the brown pixels that are present in the slide. So not all brown pixels here is going to be the CD31. So the CD31 occupies internally the lumen. So the lumen morphology is also important to determine whether they are blood vessels or not. So as you see in this picture, there is this patient is kind of responding, so that the architecture of the blood vessel changes after the radiotherapy. So it is very important to measure these dynamic changes that are happening during the therapy. And these samples are part of clinical trial CORTOC, and they are systematically collected and they are matched. We have this matched patient data before radiotherapy and after radiotherapy. And so we know that we wanted to counter for the other different cell types, such as macrophages, neutrophils, all existing in tumor microenvironment, but we are currently thinking about the integration of them at a later stage. So we give the slides to the pathologist, and the pathologist, which is Emily Jury, who is the PhD student in our lab. So we effectively collaborate with them, because it's very important that you get the knowledge of the blood vessels, because within any histology sections there could be the dead section of the tumor as well, which is called as necrosis. So you have to avoid them as well when you're collecting the annotations. That is going to be the pool of training data that goes into your training regime. So you would want to effectively collect the data that contains only the blood vessels, and you also have to exclude those regions such as background and which are necrosis, because the CD31 also stains in some of the background. So you need to give the positive representation and the negative representation in your training regions. So this is an example of positive train region. So as the previous speaker also mentioned that, so whenever you are collecting the pool of data, you cannot directly patch them and directly give them to your deep learning or parse them to the deep learning. So you have to select the positive representation, and you feed the data with the positive representation. And if you are having like 90% to be the negative data, which is the empty tiles, then it could overtrain, and then your segmentation output is not going to be very well represented in your training data. So you have to always be thinking about the model overfitting because of the negative representation that you have given. So you have to collect the positive representation and the negative representation. That takes an effort to generate this pool of training data depending on the project that you have. And as I said already, so there are much more vessel movements, the background staining, and the vessel size is not very constant, so you have to capture all these information for getting a reliable and reproducible metric as a segmentation output. So the overall workflow, so what we did is, as we know, the whole slide images are in gigapixels, and they form, we need to collect the tiles. So the tiles that we have collected are of size 2000 by 2000. From there, we have extracted the patches of size 508 by 508 for the training regions that can be passed to the deep learning network. And then we used two loss function, which is weighted by cross entropy and modified Hausdorff distance to regularize the network. And then the amount of training data from the 8 WSI that we had is 60 GB, which is 70% of training and 30% being validation. And then we assess them. And the metric that we obtained, or the features that we obtained from the histology, in future, we wanted to correlate with the novel imaging technique like super resolution ultrasound, so that we could come up with the surrogate markers for the progression of the tumor given the radiotherapy to the patients. So here, how we designed the model is, this is the traditional unit architecture with using inception encoder based on the filters being redesigned. So what we have done is, we have used one encoder and then two decoder networks. So one encoder and the decoder takes the input and the mask as the input for these ones, so that it does the segmentation task. But we generate a distance map so that they can be regressed, and then we apply the mean square errors as the loss function, and we combine those outputs to generate the segmentation output. So herein, the classical segmentation which you're using with unit or multiple other variations of unit, you only give the image and the mask. But then here we generated this normalized distance map that we feed it to the another branch of the regression, as I showed here, so which is represented in green, so which helps in refining our segmentation output, and it encapsulates automatically the variation in size and shape of these blood vessels. And for the segmentation branch, we have used the weighted binary cross entropy so that it doesn't include the borders, and it better separates the collectively represented blood vessels. And then we also used half-stroke distance so that the prediction mask and the ground truth mask, when it is closer to each other, the distance is going to be less. So this is the breakdown of the data that we have got from these, and the parameters of the network is 117,041, and then these are the 70% and 30% breakdown, which equates to 60 GB of the data. So as I said, so whenever we train any deep learning model, first we evaluate on a small tile. So, so this is the metric that I'm going to use for the segmentation output. So as you see, when you have the center of the mask, is the white region is going to be constant, and just imagine the other one is prediction. This is for demonstration purpose. So whenever the white and the pink is going to be overlapping, you will get the dice to be high and the distance to be low. So the same thing that we have followed. So you have the ground truth and the prediction in the bottom, and we generate a segmentation overlay map wherein the green pixels are the true positive, which is matching the both ground truth and the DL-based segmentation, which is part of the true positive, and those that are in the pink are the false negative, and those that are in yellow are the false positive regions. So from here, we also do some post-processing so that we reduce the false positive, because we know that the blood vessels are having some lumen. So those blood vessels in the DL segmentation that doesn't have the lumen, we exclude them so that we reduce the false positive, and then we also apply the smoothing and the filling of the output DL-based segmentation, and then we improve on the false negative as well. So this is one visualization at a tile level, generating this segmentation map overlay on these. And we used Napari tool in order to get the segmentation overlap on the overall image, because it varies so heavily, and some of the blood vessels also splits. So currently we are still working on those splits that are happening, one blood vessel being called as two, because we see that the panoptic quality is decreased. So we are trying to improve that on the same. And the dense inception that was part of a monosec challenge for the cell-based analysis, we have used, without those variation, we got a die score of 0.73, but by adding the attention map and the distance-based regularization and the post-processing, we see that the performance is increased. And these are the outputs for all the tiles that had positive representation of the blood vessel, excluding the negative regions for the 5 whole slides. So because if you adding the negative regions, because already they are part of the blue regions, if you remember the segmentation overlay, that will be always 1. So it will skew your results. So I'm showing only those regions that had blood vessels, because we know where the ground truths are. So you can exactly compute what the die scores are there for every patches that we have. So the idea is to assess the scalability and generalizability across the clinical trial that we have got, so that we will be able to measure the changes of the microvasculature to predict the long-term response to radiotherapy. And then the future is, as I said, it is going to be the contrast enhanced super resolution ultrasound, so that we get measures from there and then correlate the measures of tumor vasculature that we estimate from the histology slides as well. This is an ongoing collaboration with Imperial College. And we also think that blood vessel doesn't exist alone in a tumor microenvironment. There is also other adaptive immune cells such as T cells and B cells existing in the environment. So we are looking at expanding the generalizability of the model that is encapsulating the morphology, but also the cell level details that we can add when we are having enough of the MIF images that we acquire as a part of this trial, and other collaborations with the PhD students from CTI lab in ICR. And here I would like to use this platform to promote my previous work, which is Unmask Framework. So wherein we have studied which of the phenotypes of the breast cancer is growing into invasive. So we have collectively used multi-resolution feature engineering, and then the ecological characteristics of how epithelial cells tries to become invasive by recruiting the surrounding cells such as extracellular matrix and the immune cells that are present in the vicinity. And then now we are studying the collagen characteristics that are responsible for poor prognosis in patients as well, which is one of the posters by a PhD student, which is poster number 3. You're welcome to contact either me or my the PhD student from our lab. And this is all not possible without the collaboration and my lab. And thank you everyone for listening.
Yes, questions? We are a few minutes ahead of schedule, so don't be afraid to find one of the microphones or ask for the purple cube, whichever one you prefer.
Okay, so yeah, we have a question on Zoom. Have you tried to get a different annotator to estimate the human error and compare it with your network?
Like we had 2 or 3 iterations from the pathologist, and that's how we learned that there are some necrosis which are not part of the, so it is an iterative process that we did. So we already have seen such problems and counteracted for that. And the segmentation overlay map that I shown as a video already has a necrosis tissue. And sorry, I missed that one. Yeah, so the part of the tissue on the upper side of the slide is the necrosis, and the one in the bottom are the active tissue. So we are not taking into the account that are part of necrosis already. Thank you.
Questions in the room?
There's another one at the back.
Sorry.
Okay, sorry, yeah, go ahead.
Thanks so much. I was just wondering if you noticed any variations between staining batches on the IHC. So were there some slides that were segmented better than others, just to do with the biology?
Yes, we have seen such changes because these were collected at 2 different time points, right? So I think one of my slides also has that variation. So you could see the one that was annotated is totally different from one that went into training. So we already see that one, and the algorithm is able to counter those challenges regarding the intensity differences in different batches. So, yeah.
So sorry, how did you go about correcting for batch effect?
because the inception network automatically has the batch normalization, so I think it is already inside within the network, but we didn't do like a stain normalization or anything, because the stain normalization is totally different on a HNE and on an IHC image. So we have not used any stain normalization here, except the augmentation that we do regularly with deep learning, and then the batch normalization that is within the deep learning architecture.
We have time for one more.
Okay, one more online. Would you consider adding further markers, as CD31 alone may not be adequate for assessing neovascularization?
Currently in this project, we don't have any additional marker, but yeah, so that's why we wanted to extend that with multiplex as well, to see the whole story. So, but then we need to make this work systematically with addressing each of the TME components. So we are building it as a module, and I think at one point I'll be able to answer that question.
Okay, that's it.
Thank you. Let's thank the speaker again.